into the virus design and our focus in the lab to devise a vaccine candidate that could improve current FMDV vaccines. And you may be more familiar with our leading candidate that is based on a leaderless a virus that is highly attenuated and could be used as a seed, safe seed virus for production of foot and mouth disease. And that virus has also built-in negative marker for DIVA test determinations. So today I'm going to focus on more recent development in the lab that in this case we have tagged the capsid by either inserting or fusing Histag. In one case we just inserted at the carboxy terminal of EP1, and in other cases we have fused this Histag to the inserted in 2A and is presented as a fused 2A Histag to the capsid. So in predictions of this structure, we estimated that these Histags are going to be exposed, sticking out from the capsid in this way, either fused to the 2A or just at the carboxy terminus of EP1. And this allowed us to have a functional Histag presented on the surface that could either bind to antibodies, bind to a solid surface where nickels is presented, or to more complex molecules where nickels is also the mean of tagging to the capsid. So these viruses were characterized in vitro, and we demonstrated that the kinetics of growth of the virus in VHK cells are similar to the parental virus, but also the properties of the morphology of plaques and, more importantly, the titles are also very similar to the wild type. Also important was to determine if the antigenic composition is not affected by the presentation of the Histag at the carboxy terminal exposed on the surface, and we just measure how they are neutralized by serial arrays against the parental A24 Cruzeiro virus, and you could see that the mutants are equally neutralized. And we have also, and although not presented here, look at the profiling by using monoclonal antibodies to the GH loop and other antigenic sites, and the mutants and the wild type are equally bound to those monoclonals. Also important, if we look at the EM electron microscopy images of the wild type or the mutant virus, they seem very similar. And, but if we combine them with a nanolipid, nickel nanolipid molecules, complex with, in this case, is an adjuvant MPLA, you could see that all of a sudden we start seeing these nanolipids decorating the surface of the mutant virus presented right here, while the wild type is never really associated specifically to those nanolipids that in EM look like very disordered molecules. We measure how many of these nanolipids, and of course you have 60 possibilities on the capsid to bind these molecules. So this is very important when we, for instance, combine them with adjuvants fused to nanolipids. We have looked at some of the properties, and actually if you have ELISA plates with nickel covering it, we could just take supernattants of infected cultures from these mutants and bind to this plate, and they could easily compete it, and you have this competition presented here, where these complex nanolipid molecules could actually compete the binding to the plate of the mutant viruses. We have also looked at the possibilities to just take the supernattants of the mutant virus and pass them through columns, affinity columns, either cobalt or nickel columns, and we are able to recover on the other side in this affinity purification scheme, viruses that here we are looking at infectivity. So we were able to recover in the eluate up to 90% of the PFU of the functional virus on the other side, measured here by titration. But also important, using this elution system, we were able to eliminate the non-structured proteins and purify effectively the capsid. This wasn't really the case of, as effective as we found this elution system with the EDTA, where we still see some of the non-structured proteins in the prep. So it was important to not only look at the functionality of the capacity of these Histags to bind antibodies to be used as a support to purify the capsid effectively, but also what would be if we combine with adjuvants that can be almost spontaneously, because it's just a matter of minutes or less seconds to really make this complex with adjuvants decorating the capsid. So we formulated and tested in a mice model as a preliminary experiment, where we just took this mutant capsid, combined them with the conjugated nanolipids with MPLA, the adjuvant, and you could see by EM that we decorated this capsid with the adjuvant. And then we devised an experiment where we tested different concentration of the virus, the mutant virus alone, or the one that has been conjugated with the adjuvant by means of the nanolipoproteins. And of course, here are the controls, which you could see that in this challenge virus where we measure lethality, we see that by 2 or 3 days, the challenge caused the death of the controls animals, while we have a significant improvement on the protection conferred by the combination of the mutant virus with the nanolipid adjuvants. The neutralizing titers also were slightly significantly higher, and the viremia was reduced in the case of the combination of the mutant virus with the nanolipid adjuvants. So also important was to determine if the Histag is being detected in these animals. So we look at the sera from either animal that has been infected with the virus, with the wild-type parental virus, or the sera from those animals that were vaccinated with the complex of the mutant virus and the adjuvant. And as you could see, there is a significant difference on the ability of this serum to recognize a protein put in the ELISA plate that has a Histag. And this is a completely different protein, PCVP, that is a Histag. So we are measuring really the antibody response to the His, and that is present in the vaccine, allowing this differentiation. So in summary, I presented some data here of the development of these mutant viruses that exhibit, similar growth, kinetics, plaque morphologies, and antigenic characteristics at the wild type. By electron microscopy and by biochemical analysis, we detected that there are functional Histags exposed on the surface of these antigens, and they could also complex with more complex molecules like the nanolipoproteins adjuvanted with MPLA. The Histag also allowed one-step purification of the mutant capsids, which is an advantage when you want to also eliminate some of the non-structured proteins, and this process could be scaled up. Animals immunized with the inactivated virus fused to this complex and presented as a vaccine produce an enhanced protective immunity against FMDV compared to the virus alone. And finally, the induction of anti-exHis was also demonstrated in these animals. Finally, I would like to just acknowledge people in the lab, especially the Vendra Raid, who performed this experiment, collaboration with the lab of Teresa de los Santos, Faina, Luis Rodriguez, And also very important, our collaborators on providing the nanolipid molecules from Lawrence Livermore National Labs. With that, I would like to just answer any questions.
Thanks very much, Elizabeth. Do we have some questions? We have a little bit of time.
Elizabeth, very nice presentation. I understood very well about the Histag insertion to the capsid protein, P1, but I could not understand the one you have fused with 2A. And my understanding is when capsid is processed, 2A is cleaved by 3C. So how it will be helpful for your work, or I could not understand.
So the cleavage, when we inserted in 2A became, instead of 21 amino acid, it's longer by 6 amino acid. But we also eliminated the capacity to cleave 2A, the fused 2A. So it's presented, the capsid are presenting on the surface Histag 2A.
Yeah, do you have some data compared to the commercial oil adjuvant with your adjuvant?
I think we only tested in mice at this point. Our intention is to go into the natural host, and in those instances, we are definitely going to do comparisons with commercial available vaccines and this adjuvant being formulated with the nanolipids. Yeah, that's our next step on the characterization of these vaccine candidates.